This week our focus will be on blood. We specifically want to be able to identify all of the different components of the blood, each of their different functions, and then we will get into blood plasma, how blood cells are produced, but there are definitely going to be portions of this chapter that we are going to skip over because we are still making up some time. The functions of the circulatory system we've already talked about last chapter, but blood is how these happen, so they are still relevant in this lecture. The function is to transport oxygen to the tissues and to remove carbon dioxide from the tissues. Other nutrients are also transported, wastes, hormones, and stem cells. Blood protects by going to the source of inflammation. It limits the spread of infections and helps to destroy possible cancer cells, and also initiates clotting. We'll talk about the process of clotting in this chapter as well. It also regulates our fluid balance, keeps our pH at a stable level, and helps with the control of our inner temperature. So some general properties of blood. Adults would have anywhere from 4 to 6 liters of blood. It is considered a connective tissue, but it is a liquid connective tissue. It has cells and matrix, matrix like all of our other connective tissue, but this one's just a little bit different because it is a liquid form. So plasma is considered the matrix of the blood. And back in A&P 1, when you did cells versus matrix, it was very clear these were the cells, and then everything else was considered the matrix. Well, in blood, the plasma is the matrix. It is a clear, light yellow fluid, and within that, we see our cells. Now, within blood, sometimes the cells are called formed elements, but really they are cells or cell fragments. This includes red blood cells, white blood cells, and platelets. Within those 3 categories that I just mentioned, you can break it down even further, and so eventually there are 7 kinds of formed elements. Red blood cells, which are called erythrocytes, platelets, which are cell fragments, and then there are white blood cells, but there are 5 different types. So 5 types of white blood cells, and they are divided into 2 categories. The categories are with granules that are called granulocytes, and then or without granules, which are agranulocytes, and we'll talk about how to tell the difference there. But then the 5 different types are listed there: neutrophils, eosinophils, basophils, lymphocytes, and monocytes. It's going to be important that you're able to identify each of those blood cells just by looking at a picture. And there they all are. So the red blood cells are the easiest to identify. They look like gummy lifesavers that are red. And then those little tiny purple blobs are the platelets. Those are cell fragments. They are very tiny. You will have to search for them in the lab. And then everybody else there is a white blood cell, and it shows the different types. So looking at just the blood plasma, when we separate blood with a centrifuge, it's called a hematocrit. That basically separates the components, puts all the cells together, puts all the plasma together. And so when we're looking at someone's blood, the hematocrit can tell us if there's any kind of imbalance, not enough blood cells, or other types of problems. The erythrocytes are the heaviest, and so when they're centrifuged, that means they're spun around, and they're the heaviest, so they're going to settle to the bottom. And we would expect a hematocrit of a normal person to have 37 to 52% of the volume be red blood cells. And then white blood cells are next, and the platelets together, and they're only 1% of the total volume, so they're a very small percentage. And then finally the plasma is the remainder of the volume, 47 to 63%. It's a mixture of water, proteins, wastes, hormones, and gases. The plasma is the liquid portion of the blood. The serum is the remaining fluid when the blood clots. So when the blood clots, all of the cells are headed to the clot, and the solids are removed, then what's left is just the serum. It's identical to the plasma, except it doesn't have the protein called fibrinogen, because that is involved in the clotting process, which we will get to. There are plasma proteins in the blood plasma that are important, and these three are the ones that I need you to memorize because they will each have a role later. So albumins are the smallest and most abundant. They contribute to the viscosity and osmolarity of the blood. They influence blood pressure, flow, and fluid balance. We will have a review over viscosity and osmolarity coming up. The second plasma protein are globulins. They are antibodies. They provide immune function, and they are broken down into alpha, beta, and gamma globulins. I really just need you to memorize globulins, not the three types. We'll have enough to memorize in this chapter. And then the third one is fibrinogen. It is probably the one that we will talk most about because it does help form a blood clot. It is the protein that will transform into fibrin threads that help to form the blood clots. Plasma proteins are formed by the liver, except for globulins. They are produced by plasma cells themselves. And then we'll also see that there are some nitrogenous compounds in the blood, amino acids that are floating around. They're not part of a protein, but they are from dietary protein or tissue breakdown. So they once were part of a protein, but now they are separate. And then there's also some urea, which is the end products of breaking down reactions in the body. And those will be normally removed by the kidneys when the blood flows through there. The blood plasma also includes nutrients such as glucose, vitamins, fats, cholesterol, lipids, and minerals. But probably the most important are these gases, so dissolved oxygen and carbon dioxide and nitrogen. We've talked a lot about the oxygen and carbon dioxide, but there is nitrogen in the floating around in the plasma as well. And then electrolytes. So sodium ions, being the most abundant, makes up 90% of the plasma ions. Okay, so looking at the blood viscosity, that means that's resistance to flow. It's because that the particles are very cohesive. That means they like to stick with each other. So whole blood is 4.5 to 5.5 times as viscous as water, whereas the plasma just itself would only be 2 times as viscous as water. This is very important in circulatory function. This is what allows the blood to flow as efficiently as it does. And then the osmolarity of blood is the molarity, that's moles per liter, of the dissolved particles that can't pass through the vessel wall. And this measurement is what creates the osmotic pressure. So we want optimum osmolarity by the regulation of sodium ions, proteins, and red blood cells. So basically this osmolarity of the blood is important in keeping what needs to flow in the blood, within the blood, or other particles to not pass through the vessel wall. Blood being produced is called hemopoiesis. It is the production of blood cells, but specifically we're going to focus on those formed elements, the blood cells. Adults are producing 400 billion platelets, 200 billion red blood cells, and 10 billion white blood cells every day. So there are specific tissues where this occurs. The yolk sac produces stem cells for the very first blood cells. This is during embryonic development of a fetus. But liver stops producing blood cells at birth, and then the spleen continues to make white blood cells, or lymphocyte production. If you play this is an animation. If you have this in presentation mode and make sure you've enabled the macros, then you should be able to watch this animation about the blood cell formation. So the red bone marrow produces all of the 7 types of cells. And this is talking about which type happens in the bone marrow and which type happens in the lymphatic organs. So it's called different things wherever the blood cells are created. We will probably focus mostly on the blood formation in the bone marrow.